Now we're going to focus on the events that occur during the first 8 weeks after fertilization. The fertilized egg, now known as the zygote, will undergo a process called cleavage. Cleavage is a sequence of mitotic divisions, rapid mitotic divisions. This is where the zygote will transform, basically mitotically divide into a two-cell stage, and then from there it will divide into a four-cell stage, and eventually it will give rise to a morula. Now, the morula is a solid ball of cells, basically contains 16 to about 32 cells. And then the morula will then give rise to the final stage known as the blastocyst. So remember, the fertilized egg, its goal is to get to the uterus for implantation. But as it travels along through the process of peristalsis, it will rapidly divide by mitosis. We'll go from the two-cell stage to the four-cell stage and then to the morula phase. Now, do not forget what I said. The morula is a solid ball of 16 to 32 cells. All right? And notice that the zona pellucida is still surrounding these cells. Now, from the morula, we will end up with a blastocyst. And it is the blastocyst that will implant into the endometrial lining of the uterus. Before we have a mature blastocyst, we first have an early blastocyst. And this is basically when the morula, you know, hollows out, fills with fluid, and then hatches from the zona pellucida. So there are some cells. These cells are known as the trophoblast cells. They will secrete digestive enzymes to break down the zona pellucida and hatch out the blastocyst for implantation into the endometrial lining. So let's focus on the specific structures of the blastocyst, this implanting blastocyst. It is made of the inner cell mass. This inner cell mass is actually a cluster of small round cells. This cluster of cells will give rise to the embryonic disc that will form the future embryo. Next, we have the trophoblast cells. They are large, flattened cells that form the outer layer of the blastocyst, but they are very, very important. And here's the reason why. They provide nutrients to the developing embryo. They will form the future chorionic sac and make up the fetal part of the placenta. But the primary thing I want you to know about the trophoblast cells is this. They secrete HCG, human chorionic gonadotropin. It is a hormone secreted during pregnancy. They secrete HCG to stimulate the corpus luteum in the ovaries to continue to produce its progesterone. Now, why is that important? Well, we need progesterone to maintain the endometrial lining during pregnancy. That is why it's important, and I want you to know that. Next, we have the blastocele, also known as the blastocyst cavity. The blastocele is actually an inner fluid-filled space. Okay, so that is all that we have here. Make sure you know the three structures that make up the implanting blastocyst. Now, by the end of the first week of development, the blastocyst will come in contact with the endometrial lining. Now over here, I want you to notice here's the blastocyst. So you see the structure right here called the embryoblast. This is also the inner cell mass. So the inner cell mass has two names. It has, of course, the inner cell mass, but it's also called the embryoblast because it gives rise to the future embryo. Don't forget that. And of course, we have the outer layer made up of the trophoblast cells. Now, implantation actually is due to these trophoblast cells. Because the moment the trophoblast cells make contact with the endometrial lining, they will differentiate into two specific layers of cells. So the trophoblast cells, they make contact with the endometrial lining. They will now differentiate into two layers of cells. The first group, which are the innermost layer, we call them the cytotrophoblast, the cytotrophoblast cells. Now, these group of cells, they will help to develop the chorion. They will form the chorion and the chorionic villi. Now, I know you don't know what a chorion is at this moment, but don't worry, I will explain later. But the chorion is actually one of the extra embryonic membranes that will surround the growing embryo. It's more of a protective layer, okay? And it usually has these finger-like projections coming out of it, known as the chorionic villi. And the purpose of it is to actually help in forming the placenta, the fetal part of the placenta, which will make contact with the maternal blood for, gas exchange, nutrient exchange. Now we have the other layer called the syncytial trophoblast. This is the outermost layer. The syncytial trophoblast, these cells, they secrete growth factors and digestive enzymes that will help erode the endometrial lining and allow the blastocyst to firmly secure itself, embed itself within, you know, the endometrium. So that is the purpose of the syncytial trophoblast. So don't forget the two. And while this implantation is taking place, you will notice a pool of blood will begin to surround the embedded blastocyst. And the reason is because these digestive enzymes do end up, breaking down or destroying some of the blood vessels of the endometrium. So we will have a pool of blood, a leakage of blood all over the place surrounding the blastocyst as it implants. So please remember, this is all about implantation right now. And the implantation is spearheaded by the trophoblast cells. They're the main ones responsible for this. Okay? Once implantation is completed, the uterine mucosa will regrow itself and surround the embedded blastocyst. All right, and you can see that right here. Now, implantation is usually completed by the middle of the second week of development. However, while implantation was taking place during the second week, something else was taking place too. Now, remember that inner cell mass that we talked about, this embryoblast? Well, it will begin to differentiate. So the embryoblast will differentiate into a two-layered embryonic disc. This embryonic disc is also called the bilaminar germ disc. But the two layers that I'm referring to is the epiblast and the hypoblast. So the embryoblast, or the inner cell mass, will differentiate into two layers, and you can see that right here. So this was the inner cell mass, and here are the layers. So this is the epiblast and the hypoblast. And notice how it creates two cavities on either side. So the hypoblast is facing the blastocyst cavity right here. And here is the epiblast facing the amniotic cavity. Yes, that sounds familiar, right? So the amniotic cavity will eventually fill up with amniotic fluid, all right, which will surround the growing fetus. Okay, now let's talk about this for a second. So this is just the differentiation of the inner cell mass. Now, what is going to happen eventually is the blastocyst cavity will eventually give rise to one of the other extra embryonic membranes that will support the growing embryo. The blastocyst cavity will eventually give rise to the yolk sac. We can say the primitive yolk sac. Okay? And then the amniotic cavity, like I said, it will become filled with, so let's erase this, it'll become filled with amniotic fluid, and these epiblast cells will then form the amnion. So this is a sac-like structure that will surround the growing embryo. Okay? So this is just a differentiation, again, of the embryoblast or the inner cell mass. Just remember, there are two layered embryonic disc made of the epiblast and hypoblast. Okay? Cavities on both sides. The blastocyst cavity will eventually form the yolk sac. All right? The amniotic cavity will become surrounded by the amnion, which actually is developed from the epiblast. And the amniotic cavity will contain amniotic fluid. So as embryonic development continues, the bilaminar germ disc, which consists of the epiblast and the hypoblast layers, they will reorganize to give rise to specific primary germ layers. And this occurs in a process called gastrulation. So all I'm saying here is the two-layered embryonic disc that we talked about, it will reorganize and form 3 embryonic germ layers. And that process is what we call gastrulation. Make sure you remember that. So this usually occurs during, the 3rd week of embryonic development. It begins at the end of the 2nd week of development and continues into the 3rd week of development. Okay, and of course these embryonic germ layers will then give rise to specific structures within the embryo, specific organs within the embryo, and that process is what we call organogenesis. And that process can begin from the 3rd week and continue on to the 8th week of embryonic development. Okay, so 2 processes that I have mentioned, gastrulation and organogenesis. All right, so gastrulation, like I said, is when the 2-layered embryonic disc rearranges or reorganizes itself into 3 embryonic germ layers. And those embryonic germ layers are the ectoderm, mesoderm, and the endoderm. All right. Now, during this process, there are some extra embryonic membranes that are developed. And these extra embryonic membranes are going to support and protect the growing embryo. And I do want to go over all of them. Okay, we have covered a few already. So let's begin with the first one. I'm going to start with the amnion. Remember that the amnion, this membrane was formed by the epiblast cells. And you can see that right here. Here's the epiblast. Notice it's surrounding the amniotic cavity. Remember I told you the amniotic cavity contains the amniotic fluid. Okay, what is the purpose of the amniotic fluid? Well, it will provide a buoyant protective environment for the embryo. All right, this is why we say that, the growing embryo basically swims in water. It is true. It's the amniotic fluid. Okay, so this amniotic cavity is basically surrounded by, of course, the epiblast layer of cells here, but notice the amnion is formed from this epiblast layer of cells. So we have the amniotic membrane being created right here, and it will continue to grow. So here it is. At the 16th day of that embryo, there it is, the amnion will continue to grow. Okay, eventually it will envelop the embryo. Next, we have the yolk sac. So remember I said that the yolk sac was developed from the blastocyst cavity. So we started off with the blastocyst and it gave rise to the yolk sac. We call it the primitive yolk sac. Now, the yolk sac at this stage is the primary source of nutrient for the embryo at this early stage. But after the placenta takes over in providing, the nutrients, the yolk sac will reduce in size. And it will also serve as the first site of hematopoiesis. Yes, 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 yes. It's going to serve as the first source of blood cells. Don't forget this, the first site of hematopoiesis, right? Where we make blood cells. It's going to be the first source of the blood cells and also the first source of the primordial germ cells of the embryo's gonads. Okay. Next, we have another structure called the allantois. The allantois. The allantois, if you take a look at it right here, here it is. Not this one, but this one. The allantois, what is it? Is a little out pocket, at the caudal end of the yolk sac. So it's basically going to act as an excretory duct, a primitive excretory duct for the embryo that will eventually become part of the urinary bladder. Okay, now the stalk of the allantois and the yolk sac will help to form the umbilical cord. So I'll show you another picture of it at a later time, but at this time just remember the stalk of the yolk sac and the allantois, both of them will form the umbilical cord. And finally, I want to talk about last and the outermost extraembryonic membrane. It is called the chorion. If you have been following this lecture, you will remember that I said the trophoblast cells that we started off with when we were talking about the blastocyst at its initial stage. The trophoblast cells will give rise to the chorion. I said this before. Well, here it is now. So the chorion, if you remember, the chorion is actually made by the cytotrophoblast layer of cells that came from the trophoblast cells. Remember that? So they're the ones that form the chorion. So the chorion will act as this final protective membrane that will form around the growing embryo. All right? And it is important because it is going to create finger-like projections called the chorionic villi. There are multiple of them. There's one right there. Here's another right here. There's another. And all it is basically a means of, gas exchange. These are structures that will participate in gas exchange, nutrient exchange, waste exchange with the maternal blood. So the chorionic villi will form the fetal part of the placenta. Remember, the placenta is actually made of a fetal component and a maternal component. So the chorionic villi will have contact with the maternal blood, all these blood vessels within the endometrial lining, all of it. It will have some kind of contact where there will be an exchange of nutrients, of gases, of waste products, okay, between the growing embryo or fetus and the maternal blood. Okay, so the chorion is very important. It's the outermost extra embryonic membrane, all right, that will surround all the other extra embryonic membranes that I've talked about. And overall, do not forget the purpose of these extra embryonic membranes is to support and protect the growing embryo. Now, remember the process of gastrulation gave rise to the three embryonic germ layers. I want you to know the specific organ systems that are derived from each embryonic germ layer. So, from the ectoderm uh, germ layer, you will have structures of the skin, the integumentary system, the skeletal system, the nervous system, the endocrine system, the respiratory system, and the digestive system. Okay? And also we have, here we go, we have the endoderm germ layer giving rise to structures of the following systems. So what I want you to do, look in the notes, and I want you to know I've put them in bold for you. So go through them, and I want you to know the specific structures that um, are developed from each embryonic germ layer. Okay? And please do not forget that the process by which these structures are developed is what we call organogenesis. And it occurs from the third week of embryonic development through the 8th week of embryonic development. Okay? Now we will focus on placenta formation. Now, remember that the placenta has two components to it. It has the fetal component and the maternal component. The placenta is important for nutrient, exchange, waste elimination, hormone production, gas exchange between the maternal and the embryonic or fetal blood. Don't forget that. Now, the placenta is connected to the embryo through the umbilical cord. And you can see that right here. This is how the embryo is connected to the placenta through the umbilical cord. So what are the components of the umbilical cord? Number one, we have a single vein and two umbilical arteries. So a single umbilical vein and two umbilical arteries. And these structures are suspended within a gelatinous space. And this jelly substance is what we call Wharton's jelly. All right. Now, I want you to keep in mind that the chorionic villi that I was talking about that extend out of the chorion, here's the chorion, you can see that. So this is the amnion, right? This is the amniotic fluid in here. Of course, the embryo or fetus isn't here at this time. They took it out for you to see the full structures. But this is the placenta. So you can see the part of the placenta right here. So the placenta, like I said before, is actually made of the chorionic villi. Here they are. That's the fetal component. And then it is made of the decidual basalis. The decidual basalis is basically the deepest layer of the endometrium. It's the deepest layer of the endometrium. And the decidual basalis and the chorionic villi, they come in contact with each other. Right here, you can see that in this zoomed in picture. Right here, you can see that right here. They come in contact, and this is where all the exchange, gas exchange, nutrient exchange, this is all where it occurs. This is where it will take place. Okay, so the placenta itself is formed by the decidual basalis of the endometrium and the chorionic villi of the fetus. Okay, so please remember that. All right, now there is another part of the endometrium. This is the part of the endometrium that will bulge out into the uterine cavity. We call this the decidual capsularis. So the decidual capsularis is this part. So this is more of an outer layer of the endometrium that will bulge out into the uterine cavity as the embryo grows into a fetus and the fetus, increases in size. So here's a picture that kind of summarizes all the extraembryonic membranes that will grow around the developing embryo and eventually the fetus. Here they are. Remember what I said, the outermost layer was the chorion. You can see that right here. After that, we have the amnion, the amniotic sac. There it is. And of course, we have, oh, I promised that I would show you the umbilical cord. So I did say the stalk of the allantois and the yolk sac creates the umbilical cord. And you can see that right here. This is what I was referring to when I said the stalk of both of these structures will form the umbilical cord. And then, of course, you can see the placenta here. So we have the deepest layer of the endometrium, the decidual basalis. This is it right here. This is the decidual basalis, the deepest layer. And notice how it makes contact with the chorionic villi, right? That extends out of the chorion. All right. So these two will form the placenta. Now, during pregnancy, the placenta will be responsible for certain hormones. We've already spoken about the first one, which is HCG. Like we said, the placenta will take over producing HCG. Remember, HCG initially was produced by the trophoblast cells. They made that, but the placenta will eventually take over. And remember, the purpose of HCG is to cause the corpus luteum to continue to make progesterone, which will then help to maintain the endometrial lining. Another hormone is human placental lactogen. So this is just to prepare the mammary glands for milk production. All right. Now, after pregnancy, prolactin will take over. Remember, prolactin helps to stimulate milk production. This is after pregnancy. All right. And oxytocin will help to promote milk ejection. Now, another hormone produced by the placenta is relaxin. Now, during pregnancy, this hormone is secreted. It will delay the onset of labor contractions during pregnancy. However, the closer we get to the delivery date, towards the end of pregnancy, it will then promote the rupturing of the membrane surrounding the fetus, and also it will promote the dilation of the cervix, okay, and also cause the softening of the pubic symphysis. So basically relaxing the pelvic, articulations to increase flexibility for childbirth. Okay, so relaxing during early pregnancy, its job is to prevent any onset of labor contractions. But towards the end of pregnancy, it will begin to now prep the body for childbirth. Okay. Now, of course, the placenta can also produce progesterone. It can. And also estrogen. So all of these hormones, especially estrogen, the later we get into pregnancy, during the later stages of pregnancy, estrogen levels will begin to increase. And the reason is because it will help to stimulate labor and delivery. Now, progesterone, like we said before, it is very important because it will help to maintain the endometrial lining during pregnancy.